are pinched in the middle. And if you think about it, that makes sense to be pinched in the middle. Red blood cells are little bags of hemoglobin, and hemoglobin carries oxygen. You want the oxygen to be able to diffuse in there to the hemoglobin very easily. With your red blood cell pinched in, rather than being a sphere, there's not any great distance anywhere from the outside to the inside. So it makes it very efficient for diffusion. Now, this also gives your red blood cells some flexibility, being shaped like this. You know that if you go to Wet n Wild, or you get in a tube at the springs, or water park or whatever, if you hit the wall of the side of whatever you're going through, what does the tube do? It just bends and bounces back. Red blood cells go through capillaries pretty much one at a time. Okay? The capillaries are very, very small. Okay? And they have to bend a little bit, okay? And they bump into each other. Okay? So this shape is not only efficient for oxygen diffusion, it also gives the most strength and flexibility to your little red blood cell. One of the things that is wrong in sickle cell anemia is that the shape won't stay like this, okay, under low oxygen conditions. And so the cells sickle, okay, or sometimes they will just plain old break. Okay. There are other diseases similar to sickle cell anemia, the thalassemias, okay, and in that case, the red blood cells also assume weird shapes and break. Now, if you have little points on your red blood cell, okay, do you think that makes it easier or harder to get through capillaries? Much harder. So one of the problems with thalassemia and sickle cell anemia is that the blood cells, rather than going through the capillaries, they tend to clog up the capillaries. And then the tissues downstream can't get enough oxygen, okay, which makes it painful. Okay. In addition, pointy, stiff red blood cells can't flex properly, and they're more likely to break. Okay. And then you have less red blood cells. So, and that makes it even less likely that the tissues are going to get enough oxygen because you have fewer red blood cells overall. Okay, so the shape is important. Now, when you look at red blood cells on a typical H&E stain here, you can see that each of them has what's called a central pallor. Okay, that's because they're pinched in the middle. The other thing that you can notice when you look at this is that you don't see any organelles on the inside. Okay. Red blood cells, mature red blood cells, do not divide. Okay. And they don't repair themselves. Many other types of cells divide and they repair themselves, okay, but red blood cells don't. Okay. Once it is a mature red blood cell, okay, if it gets trashed, it's trashed. So red blood cells have a typical lifespan of about 120 days. Let me check that picture is right. 3 to 4 months. So you have to be continuously producing red blood cells, otherwise you're going to wear out all the ones that you have and not have any. Okay. Red blood cells are produced in the bone marrow. Red blood cells are produced in the bone marrow.
Bone
in the bone
marrow.
marrow. Okay, now, hemoglobin, red blood cells function as little bags of hemoglobin. Hemoglobin is what binds and picks up the oxygen and carries it. Oxygen can travel just diffused into the plasma of the blood, but that's not a very efficient way of carrying oxygen. Hemoglobin is the most efficient way of transporting oxygen to where it needs to be. Hemoglobin consists of 4 bundles of protein, and each of those little bundles of protein has an iron molecule in the middle of it. The iron atom in the middle, ion, the iron ion in the middle is what attracts and holds the oxygen. So each hemoglobin is capable of holding on to 4 oxygen. The portion that contains the iron is known as the heme portion. The portion, the protein glob around it, is known as the globulin portion. Iron will change color in association with oxygen. So when you have oxygen present, you have a bright red color. When you don't have so much oxygen present, you have a more bluish color. So oxygenated blood has a bright red color because of the change there with the heme. Deoxygenated blood has a bluish color because of the oxygen not being associated with the heme. Now, sickle cell anemia and thalassemia both involve mistakes in the production of the globulins. They involve mistakes in the production of the globulins. The globulins are made up of about 287 amino acids, okay? And in the beta chains you have 287. With sickle cell anemia, just one of those amino acids is incorrect. If you look back at the DNA, just one base pair is incorrect. So you have one mistake in the DNA that leads to one incorrect amino acid out of 287, and that causes the beta chain to dysfunction and change the shape of your red blood cell so that it sickles. And then you have the consequence of sickle cell anemia. You actually have a different amino acid involved with thalassemia, but you have a similar sort of situation. Now, malaria has several different phases to it. In one phase of malaria, the protozoa invades red blood cells and grows within the red blood cell. And then the protozoa bursts the red blood cell and goes out into the blood and attacks other red blood cells. So when you have hundreds of malaria organisms attacking hundreds of red blood cells, and then they burst those red blood cells and they all go out at the same time, then they get thousands of red blood cells. And they go out again, they break the red blood cells, go out again. Okay? You have a recurring inflammatory reaction that goes on. So with malaria, you have recurring fevers that go on. You also end up, among other things, with huge anemia because you're destroying red blood cells. Now, with sickle cell, you're also destroying red blood cells. So you would think that sickle cell would be worse with malaria. However, the protozoa has a harder time invading red blood cells that carry the sickle cell gene. The malaria organism has a harder time invading red blood cells that carry the sickle cell trait. Now, you have two genes for each characteristic. So you can have two normal genes for your globular protein. You can have a sickle cell gene and a normal gene. In that case, you would have sickle cell trait. Or you can have abnormal sickle cell and abnormal sickle cell, in which case you will probably have the disease sickle cell anemia. Okay? What happens is the people with sickle cell anemia tend to die young from sickle cell anemia. The people with completely normal hemoglobin tend to die off young from malaria. So who tends to survive and breed? The hybrids, okay? Those who have sickle cell trait and a normal trait. Okay? So this is why you have so much sickle cell trait in that population, because the ones that were normal died from malaria, the ones that had the disease sickle cell died, but the ones that just had one gene survived. Okay? You have a similar situation with thalassemia. You see thalassemia in the Mediterranean, okay? Italy, Palestine, places like that. Okay? They also had malaria. Okay? So people of Italian descent in particular, or sometimes Northern African descent, will develop thalassemia. Okay? And it's the same sort of situation. Now, sickle cell anemia and thalassemia are called consumptive diseases. They consume red blood cells at an abnormally high rate. Okay? Malaria consumes red blood cells at an abnormally high rate. So let's look here at how red blood cells are produced. Red blood cells are produced in the bone marrow. Now you already know that because you know that inside of bones you have red marrow. Okay. Red marrow is where the blood producing cells reside. And by far, the largest number of cells that they produce are red blood cells. But all of the formed elements, red blood cells, white blood cells, platelets, all of the formed elements are produced in the bone marrow. And they are actually produced from the same stem cells in the bone marrow. If your body needs red cells, it will take a stem cell and differentiate it into a red blood cell. If it needs white blood cells, it will take a stem cell and differentiate it into a white blood cell, or a platelet, whatever it is needed. So what tells the stem cells to produce red blood cells rather than something else? Okay. There are two things. The first one is erythropoietin. Erythropoietin is produced by the kidneys. Now, organs have personalities. Organs have personalities. The kidney is a picky, fussy little organ. if the kidney does not get enough blood, or if the kidney does not get enough oxygen, either one, the kidney will take steps. It will take steps to increase the blood pressure, for example, and we're going to see some of those in a couple of chapters. Another step that the kidney will take is it will release the hormone erythropoietin. Erythropoietin goes to the bone marrow and says, hey, crank out some red blood cells. Now, erythropoietin is a drug that's available. Sometimes erythropoietin is used, for example, with cancer treatments when the bone marrow is kind of trashed. Erythropoietin is also used as a performance-enhancing drug, and it's considered cheating. With more red blood cells, in theory, that would make you have more stamina. Blood doping is a little bit different. Blood doping is when you take red blood cells and store them, wait until your body has replaced them, and then put the extra red blood cells back in. That's also considered cheating. Now, the problem with taking erythropoietin and the problem with blood doping is how much of your blood is supposed to be red blood cells? 45%. Okay, if you get much beyond that, you start turning your blood into sludge. Okay, it makes it harder to push the blood around your body. Makes it more likely to clot. Makes it more likely that you will develop strokes. Okay, so EPO is not a good idea. Okay. Lance Armstrong to the contrary, it's not a good idea. Now, there's a physiologic way to do the same thing. If you've noticed, the Olympic training facilities for the United States, you know where they are? Anybody know?
Montana, Colorado.
They're in the Rockies. And why? Because that's where many of the high mountains are in the United States. If you go up to high elevation, the air has less oxygen. Your body will respond to that by producing more red blood cells. And that's not considered cheating. Okay. All right, so in your bone marrow, your stem cells turn into erythroblasts. You know that blasts are cells that are either immature or cells that are rapidly reproducing. Your erythroblasts become reticulocytes, and then they eventually are released as red blood cells. Yeah.
So in any situation where there's less oxygen in the air, will your body produce more red blood cells?
Yes. Okay. All right, now how many red blood cells do you have? Okay, you have about 5 million per cubic millimeter. Okay, that's a number that I want you to know. You have about 5 million per cubic millimeter. It doesn't really matter at the end of the day how many red blood cells you have if you don't have hemoglobin in your red blood cells, right? Right?
Yes.
So your hematocrit and your red blood cell count give you an idea of whether you have the equipment to transport oxygen, but they don't actually tell you the oxygen carrying capacity, okay? Because red blood cells are supposed to have hemoglobin in them. Sometimes they don't have enough hemoglobin in them. So the hematocrit and the red cell count give you an idea of what the carrying capacity should be, but it doesn't tell you the carrying capacity exactly like the hemoglobin measurement does. Okay? So about 5 million red cells per cubic millimeter. Now, the other hormone that acts on red blood cell production is testosterone. So many guys will have red counts that are higher than women. They may have a 6 million red count versus a woman may have 4 million. Okay. Part of it is that she's smaller, and part of it is that he has testosterone, which is stimulating the bone marrow. Okay. In addition to that, men tend to have a higher hemoglobin. Okay. When was the last time you saw a woman with a hemoglobin of 16 grams per deciliter? Okay. I mean, that's almost unheard of. Okay. Now, the normal range of hemoglobin that I want you to know is 12 to 16 grams per deciliter. 12 to 16 grams per deciliter. If you don't have a hemoglobin of at least 12 grams, they usually won't let you donate blood. And I usually don't, so I go in and say, here, before you fill out all the paperwork, take my hemoglobin, see if I've even got enough to be doing this. Okay?
Woman's normal. I mean, I'm like 14-ish usually.
Okay. Somewhere in the neighborhood of 12, okay, is normal. So if you have somebody that's 14, okay, good.
And that's your
Okay.
hemoglobin?
If you have somebody that's 11, and that's just where they stay, okay, you may not be able to get it up much higher than that, okay,
Why would that happen?
because of less testosterone. Okay, now, during pregnancy, you have a whole bunch of things that change with your hemodynamic system, okay? There's dilution, physiologic dilution that we're going to talk about in repro. So, is a hemoglobin of 10 normal? No. Is a hemoglobin of 11 normal? No. Okay. But being technically abnormal doesn't necessarily mean you're going to do something about it right away. Okay? And nowadays, they will, in intensive care units, for example, they will often let people's hemoglobin go down into the 7 range if they have to, okay, before they give blood. When I was in nursing school, if your hemoglobin dropped to 10, they were running blood. Okay. Now, there's so much problems with the blood supply that they've realized that they can address that in other ways, okay, and maybe not have to just, pump in blood, okay, immediately. Okay. So we'll talk more about that as we get down the road. Okay. So testosterone, erythropoietin. Now, I'm not going to ask you much about the specific stages in the development of red blood cells. I'm not going to ask you much about the specific stages, but I want you to notice here what happens. When you start out with your erythroblasts, they look like normal cells. Okay? They have a nucleus, they have an endoplasmic reticulum, they have cytoplasm, and they have a normal cell. As you continue along the developmental pathway here, as they continue to differentiate, you start to lose things. Okay? The organelles are tossed or they go away. Eventually, when you get to the normoblast stage, the nucleus is tossed. Okay? Remember, I said that a mature red blood cell is a bag of hemoglobin. Okay? It's not able to repair itself because it doesn't have a nucleus with the instructions. It doesn't have an endoplasmic reticulum and ribosomes and all that stuff for making proteins. Why? Because it tossed all of that as it went through the developmental process. Now, right before red blood cells are released from the bone marrow, you sort of have the teenage form of red blood cells, okay? The reticulocytes. The reticulocytes usually still have some endoplasmic reticulum in the cytoplasm. So they look different. They look like they have little spots in the cytoplasm. Okay? Under normal conditions, reticulocytes are kept in the house. They're kept in the bone marrow until they're completely mature, and then they're released. But what do you suppose happens when you have sickle cell anemia or thalassemia or malaria, or a person has bled out all over the floor and they don't have enough red blood cells circulating? They send in the teenagers, okay? The reticulocytes are released. Okay? So, on a complete blood count, okay, you will have a red blood cell count, you will have a hemoglobin, you will have a hematocrit, and you'll usually have a reticulocyte count or a retic count on there. And that gives you an idea of how fast your red blood cells are getting trashed. Okay? Are we having to use the teenagers? Okay. All right, now, the bone marrow needs several different things to produce red blood cells. Your stem cell is going to be converted into a very specific type of cell. A bag of hemoglobin. So what do you need to make hemoglobin? Well, you need iron. Okay. And you need iron in the ionic form. Now, you guys have all seen iron in the iron form, right? You've also all seen iron in the ionic form. What is iron in the ionic or the oxidized form? Rust. Okay, which is also red. Okay. Iron is only absorbed in the ionic form. Okay? So you can't eat nails and have the iron absorbed. So if you cook in cast iron pans, though, can that increase the amount of iron available for you to eat?
Yes.
Yeah. Why? Because number one, they rust, and number two, the cooking process will help to ionize some of that iron. Iron is also absorbed better in the presence of vitamin C. One of the best sources of ionized iron is green leafy vegetables like spinach, collards, and things like that. Many people put lemon, which has a lot of vitamin C, on their spinach and collards and so forth, which makes the iron easier to absorb. Another place where iron is easily obtained is from grapes and raisins. And also, interestingly enough, from onions. Okay. So where did people get the idea that you needed to eat blood rare steaks to get iron? Well, their taste buds, and also blood rare steaks have got blood running all over them, and you're eating the blood. Okay, so that was the idea. Okay, you're eating the hemoglobin, so you don't have to make it, or you have the component parts to make it. Okay, the problem is iron is absorbed less efficiently in the presence of fats and in the presence of proteins. Okay, so can you fix anemia by feeding somebody blood rare steak? Yes, but there are better ways to do it. Okay? Now, iron is stored in the liver, as Christina brought up. It's also stored in the bones. Okay. And one of the reasons why it is almost always better for babies to be spaced out is because the baby not only pulls from the mother's calcium stores, the baby pulls from the mother's iron stores, and from the fatty acid stores in her brain.
Is that why they say the pregnancy brain?
We don't know exactly why you have pregnancy brain, but it might be. Okay. All right, so iron absorption from the intestine. Iron is the critical component of heme. The rest of it is globulins, which are proteins. To produce those globulins, you need folic acid and you need vitamin B12. Folic acid also comes from fruits and vegetables, and it's a water-soluble substance, which means it's very easily washed out of the body. So if you only eat a tomato slice every third Wednesday, and that's all the vegetables you ever eat, you're probably not going to have enough folic acid. We're going to see down the road that a lack of folic acid not only contributes to anemia, it also contributes to neural tube defects, which is why it's a big deal for childbearing age women to have enough folic acid in their diet. Vitamin B12 is easiest to acquire from animal sources, including dairy. But if you are careful, you can also get vitamin B12 from plant sources as well. But you have to know what you're doing. Okay. The other problem with vitamin B12 is that it requires help to be absorbed across the gut. It requires intrinsic factor from the stomach. So the stomach has to produce intrinsic factor, and then when the whole thing gets down to the small intestine, the intrinsic factor allows the vitamin B12 to be absorbed from the small intestine. So if you want to have globin molecules that are put together correctly, you need to have enough folic acid and enough vitamin B12.
So when you take an injection of B12, how do you absorb that?
It bypasses the gut entirely, and it's just absorbed directly into your bloodstream.
So, what are the plant-based ways you can get B12? You said it is possible.
There are yeasts, and I believe that there's at least one grain that has B12. I'd have to look it up. I forget.
Okay.
Off the top of my head.
Green.
Jess, if you got a browser open, look it up. Okay, I'm going to go on, and she'll look it up.
Okay.
Okay. All right. Now, if your hemoglobin is not put together properly, or if you don't have enough building blocks to make hemoglobin properly, your red blood cells are not going to have as much, right? Right?
Yeah.
Okay. Your hemoglobin level will be low, and your red blood cells will not have as much hemoglobin in them. On your complete blood count, there are two measurements: mean corpuscular volume, or MCV, and mean corpuscular hemoglobin concentration, or MCHC. Both of those tell you how much hemoglobin is in your red blood cells. Okay? If you don't have enough iron, you typically have very small, pale red blood cells, and that's called microcytic hypochromic anemia, and it's usually from iron deficiency. Microcytic hypochromic. If you are B12 deficient, or if you are folic acid deficient, you will also have pale hypochromic cells, but they will be larger. They'll be megaloblastic. So, or megalocytic hypochromic anemia. So that's the reason why those numbers are on there. If somebody's anemic, it gives them some information about why they might be anemic. Okay. Are they burning through red cells faster than they should be, which would mean their retic count would be high? Are they iron deficient, which would mean that their MCV would be low? Okay. Or are they B12 deficient, which would mean that their MCV and their MCH would be bleh. Their MCV would be big, okay? And in both cases, their MCHC would be low. Okay? Does that make sense?
Mm-hmm.
It allows you to distinguish between the different types of anemia. Now, red blood cells circulate for 120 days. Okay, that's about how long it takes for them to wear out if they are normal red blood cells. They circulate for 120 days, about, if they are normal. Okay. Eventually, they start to wear out. And in this case, the liver and the spleen remove the damaged, worn out red blood cells from the circulation. That's why you don't have red blood cell fragments clogging up your capillaries, because the spleen gets rid of them. The liver also is capable of getting rid of them. Okay. Now, your body is thrifty. Your body will take apart your old red blood cell and it will recycle the iron. Okay. Your body will take apart the old red blood cell and it will recycle the iron. Okay.
Why does iron iron supplements make you constipated?
Because they slow down transmission in the intestine. All right, so your body recycles the iron. Okay. There are other breakdown products as well from your hemoglobin. Okay. Those other breakdown products are biliverdine, which is green, okay, and bilirubin, which is yellowish. Your body gets rid of bilirubin through the liver by making bile out of it. And then the bile gets dumped into the intestine and the excess can go on out with the feces. When you have liver disease, that process does not take place properly and the bilirubin can back up in the blood. Also, when you have an immature liver, that process may not occur very effectively, and you may have bilirubin backing up in the blood. If you have too much bilirubin in the blood, what do you see? Yellow jaundice, okay? And what do you do about it? Okay, you fix the initial problem. Okay, if a person has hepatitis, you deal with the hepatitis. Okay, if it's a baby who has an immature liver, what do you do?
Is there a strong?
Isn't it
Okay.
called biliary something or a tree?
Biliary atresia, you
Yeah.
call it. That's a
Yeah.
mouthful.
You were there though.
I'm making this. I'm doing the simple thing here. Okay.
Sorry.
All right. Okay, so with babies, okay, you are assuming that their liver is going to mature and catch up. Okay, but in the meantime, jaundice can cause bilirubin to be deposited in the brain and other tissues where you don't want it. Okay. The good thing is that bilirubin can be broken down by exposure to ultraviolet light. Okay, so they will put preemies, for example, that get jaundiced under Billy lights in their little incubators. Okay. Or a lot of breastfeeding women will sun their babies. Okay, that used to be a thing. You used to sun babies. Did you know that? Okay. And there was a couple of reasons for that. Not only it diminished jaundice, but it has an effect on their vitamin D production and how likely they are to get sick. Okay. Now everybody's afraid to expose their babies to the sun unless they have them slathered in sunscreen. Okay. Should you burn your babies? Oh, of course not. Okay. By the same token, okay, the system works this way. Okay. All right.
Veganhealth.org on B12 is somewhat inconclusive. They said the only place that they've really improving it has been in nori, which is seaweed. But even that, there's just a lot of inconclusive scientific research, basically, that they're saying there's certain things that it's been claimed to be accessible in, but it's really not necessarily been proven. So it sounds like I think there's inconclusive studies
Okay.
that have been done.
So, what's the take-home message for somebody who's a strict vegan? They either need to choose what they eat very carefully and choose foods that are supplemented with B12, okay, like brewer's yeast is often got B12 in it, or they need to take a supplement of, a regular supplement, shots or the stuff that is absorbed in the mouth, okay.
Yeah, that's what they're saying. They're saying really you couldn't rely on these alone, these food sources,
Yeah, it's
for
not
your vitamin
recommended.
B12. Yeah, it's
Well,
not
what
recommended to.
Now, most people in the United States are not in any danger of being B12 deficient. Why?
Because
It's
it's
gonna
gonna
be
fortified drinks.
No. Because a typical American diet contains animal products, some way or the other. Okay, eggs and milk and dairy products,
Oh,
and
right.
fish and meat and whatever. Okay, so usually in the United States, when you have somebody who's B12 deficient, it's because their stomach is dysfunctional, not because they're not getting enough of the vitamin.
How much do you
However, that's one reason why, especially with pregnant people, you ask how they eat. Okay? And in a typical OBGYN's office, when they hear that somebody's vegetarian, a lot of times they flip out. Okay? And the iron and the B12 is the reason why. Because a lot of vegetarians go off and be vegetarian for, how do I put this, silly reasons, okay, and don't actually educate themselves to how they eat.
Yeah.
Okay? And so they're not getting the nutrients secondhand from the cow, okay? And so then they're not getting the nutrients at all because they're not eating properly. Okay? Does that make sense? Somebody had a question back there.
I just want to know how much B12 do you need?
Oh, it's in international units, micrograms. It's not very much.
Is that what international units are, the micrograms?
No, international units have to do with. Just saw this the other day. Bacteria. How much bacteria you need for certain things. I have never bothered to learn the amounts of most nutrients. Okay, with iron it's 18. It's pretty easy to remember because the nutritional authorities argue about climate change.
B12 especially, some countries think it should be higher, we think it should be at 1,000.
Right.
Is what it is.
So it's easy to look up.
No, like when they run yours, it should be, like I take 1,000 a month. You know, like micrograms.
That's easy to look up with the current recommendations. All right, now this is the picture that your book has. It's a little bit more complex, but it basically says the same thing. Okay, iron is stored as ferritin or as hemosiderin. Hemosiderosis, okay, is when your body is storing too much iron. Where's the iron supposed to be stored? In the liver and the bones. Okay. Now, if you have too much iron, you're going to end up having too much of it in the bones and too much of it in the liver, and then your body's going to start sticking at other places where it really shouldn't be. Okay? And when you put metals in places where they shouldn't be, it causes weird, funky diseases. Okay, think house. Everybody remember house?
Mm-hmm.
Metal storage diseases are weird. Okay, whether it's copper, iron, whatever. Hemosiderosis was considered to be one of those weird iron storage diseases that nobody ever saw. And that was before we hit modern Western society and everybody ate too much meat and had too much hemoglobin coming in and too much iron coming in. And then they realized there was a subset of the population who was storing all that iron. Okay? They tend to be Northern Europeans. Now, back in medieval times, there were several different episodes of what they think was bubonic plague, wiped out between 25% and 30%, depending on which historian is guessing what. Totally disrupted society. Okay. Some people survived it, some people didn't. Have no idea at this point why some people survived it. They think that it may be because people with hemosiderosis could hang on to their iron very efficiently. It turns out that many bacteria like plague they don't care that they're eating you. They care about getting at your iron. And if you can lock up your iron, that organism is not going to be as effective in infecting you. Okay? So it's thought that the reason why you have so much hemosiderosis in that population is because these were the people that actually survived the plague. Okay, we talked about that.
It's 2.4 micrograms a day.
That's what B12 is.
And 2.6 for pregnant and 2.8 for breastfeeding.
All right, now your nutrition teacher may make you memorize that. I'm not going to make you memorize that.
How come some people are still like 1,000% of the daily needed value?
Because they're selling you something.
AnP a lot of it.
But if you wouldn't though, right, if you got the right amount?
Yeah, if your body is able to absorb the right amount. And then you get into that whole thing that you can do about whether you actually can absorb the form they're selling you. And then you get on top of that the wrinkle that things are absorbed better depending on what they're absorbed with.
Yeah.
Okay? And sometimes we don't know what components of plants in particular are helping nutrients to be absorbed. We know what the nutrient is. We don't know what the other components in the plant that are helping it being absorbed. Resveratrol and all this kind of stuff. Lutein. These other things.
Doesn't iron, you can't absorb iron if you take the calcium because they love each other.
Well, yeah.
And if
Iron
you
and
look at a lot
calcium.
of multivitamins, if you don't look on the back, they have
Yeah,
iron
calcium.
and
Calcium.
Calcium.
yeah, so you're not getting either one of them.
Okay. And again, dairy products with iron, okay, doesn't usually work out well because of the calcium and the protein content of it.
Yeah.
Mm-hmm.
All right.
You have to take it 2 hours, at least 2 hours apart.
So you eat your spinach and then 2 hours later you can drink your milk.
Yeah.
I can't drink milk.
So,
Now, there's a lot of people that are lactose intolerant. What does that tell you about the absolute necessity for you to have milk to survive?
Yeah.
Right?
Almond milk is fortified with calcium.
Yeah, milk's fortified with calcium. It's also fortified with vitamin D. All right. You have to be careful because there's a lot of orange juice out there that's fortified with calcium. Okay, you expect to get vitamin C from orange juice. So if you're eating raisin cereal that's fortified with iron and orange juice for breakfast because you're trying to fix your hemoglobin, okay, and then you put milk on it, okay, or you take orange juice that is fortified with calcium, okay, you're screwing up what you're trying to do.
I think a lot about absorption. I wish I understood, anyway, looking
We'll get
forward
there.
to digestion.
You won't get there. And quite frankly, there's less known about
I
gastrointestinal
know.
absorption than probably any other aspect of physiology.
I know.
All right. Okay, stem cells. Stem cells in the bone marrow are called hemocytoblasts. Stem cells in the bone marrow are called hemocytoblasts.
Hemocytoblasts.
Now, if you have too much of a particular type of cell, it's called a cytosis. If it's a cancerous process that is causing, wait a minute, okay? If it's a cancerous process that is causing you to have too many cells, it's called anemia. So leukemia is a cancerous uncontrolled production of white blood cells, or leukocytes. When you have not enough, it's called apenia. So, for example, if you have leukocytosis, you would expect to see what? Too many white blood cells. That may be because the person has an infection, or it may be because the person has leukemia. Okay. If you have leukopenia, what's the problem? You don't have enough white blood cells. Now, the cancerous conditions cancer has two characteristics. You have an overproduction of abnormal cells. You have an overproduction of abnormal cells. I used to not understand why patients with leukemia died of infections. Like, well, wait, if they have all these white blood cells, what's the problem? Well, it's because they're abnormal white blood cells that don't work. Okay. So all the different types of leukemias can be broken down into where in the process they left the rails, where in the process they started being screwed up. Okay? So you have acute lymphocytic leukemia, you have chronic lymphocytic leukemia, you have acute myelocytic leukemia, and acute or chronic myelocytic leukemia. And then you have the lymphomas, which we'll talk about more when we get to the immune system. Okay? So, you take your hemocytoblasts or your stem cells, and then you can make what you need out of them. Okay? The one that we have talked about so far here is the production of red blood cells or erythrocytes. Now, next week we're going to talk about thrombocytes in more detail, but one of the things that your stem cells can produce are big, huge cells called megakaryocytes. The megakaryocytes break apart into cell fragments called platelets. So this leaves us now with talking about white blood cells. Now, white blood cells in general are divided up into granulocytes and agranulocytes, and I hate it. A granulocyte is supposed to have granules in the cytoplasm, and they do. Agranulocytes don't have granules in the cytoplasm, so you would expect, if you were a normological person, to see nothing in the cytoplasm, right? And that's not true. Okay. So you have granulocytes and you have agranulocytes. Now, you also have teenage cells as well with white blood cells. As your white blood cells develop, the nucleus changes or the cytoplasm changes. The nucleus changes or the cytoplasm changes. It's easiest to see the nucleus change over here with the granulocytes. We think of nuclei as being like a round ball, right? That's what the nucleus looked like when we looked at it in chapter 3. In these teenage cells, the nucleus it looks as if it's getting curved, or it looks like it's getting smashed and flattened out. And sometimes it looks like it is being pinched off or segmented. So with your granulocytes here, your teenage cells are called bands, or SEGs, which stands for segmented cell. Just like with red blood cell disorders, where you're trashing a lot of red blood cells and you end up with a lot of reticulocytes, if you have a raging infection and you're trashing a lot of white blood cells, you tend to end up with a lot of bands and SEGs in your white blood count. So, a white blood cell count, a normal white blood cell count, is below 10,000. Now, different textbooks will give you a slightly different range. Okay, one that I happen to be looking at here says 4,800 to 10,800. Again, to me, it's easier to remember one number. Okay? Around 10,000. Okay? If it's way above 10,000, you've got an infection or you've got leukemia. If it's way below 10,000, okay, you may have a problem or you may not. Okay? But around 10,000 is what the normal white blood count is. Okay? So the first thing your white blood count or your white blood count with differential gives you is the count of all of them. When you ask for a white blood count with differential, it tells you the number of each different type of white blood cell, and it tells you whether you have bands or SEGs and how many of them. Now what we're going to see is that each of these different types of white blood cells has a specialized function. So if you have an increase in one of these types down here, what does that tell you? You have a specific problem. That's why you do a white blood cell differential. It gives you a tip-off to what general category your problem is in. For example, neutrophils here are specialized for taking out bacteria. Lymphocytes are specialized for taking out virally infected cells. There are two diseases common in university student populations that cause sore throats with a bunch of gunky exudate all over the throat. One of them is mononucleosis and the other one is strep throat. Strep throat is a bacterial infection. You would give antibiotics for it. Okay. Mononucleosis is a viral infection. You don't give antibiotics for it because you can have your person break out in rash. And furthermore, if you give antibiotics for strep, your person is usually over it in a couple of days. With mono, they need a whole stack of excuses for their classes because they're going to be out for a while. Okay? So it's two different ways of dealing with the process. Now, back in the day before we had rapid strep tests, what we used to do is send them down to the lab and have the lab do a quick white blood cell count with differential. If the neutrophils were high, then we knew the person had strep throat. If the lymphocytes were high, then we knew the person had mono. So that's how you do that. Okay, so some comparison here to your counts. Okay, we said that by far you have far more leukocytes in the blood. Okay. Excuse me, you have far more erythrocytes in the blood than leukocytes. And your leukocytes are divided up into granulocytes and agranulocytes. Now, those two categories are further divided up into neutrophils, eosinophils, basophils on one side, and then lymphocytes and monocytes on the other side. The biggest two categories are neutrophils and lymphocytes. The biggest two categories are neutrophils and lymphocytes. upstairs in the white notebook that has pictures in it, those tissue pictures, there are also pictures of blood cells. And I might show you different blood cells and ask you to tell me what kind of blood cell it is. This blood cell here is called a neutrophil. Okay. It's also called a polymorphonuclear cell. Nobody wants to run around saying polymorphonuclear cell all day, so it's also sometimes called a PMN or a poly. So polymorphonuclear cell, PMN, poly, and neutrophil, they all refer to that critter right there. How do you tell that what you're looking at is a neutrophil? Well, looking at that, what's the first thing that pops out at you?
The granulocytes.
It's got a weird looking nucleus. Okay. It does have granules in the cytoplasm, but its nucleus is where? Okay. The morphology or the shape of that nucleus doesn't look normal, right?
Mm-hmm.
It looks like it has bunches of different shapes. If you looked at another neutrophil, it wouldn't look exactly like that. Its nucleus would have a different shape, okay? All looking pinched, okay, like that. Okay? So with neutrophils, you have multiple shapes for the nucleus, polymorphonuclear, and the granules stain neutral, which is how they get the name neutrophil. Okay, the next type of granulocyte is an eosinophil. There are far, far fewer eosinophils than neutrophils. And what's the first thing that pops out on you there? Okay, you got granules, but the granules are really, really dense. Okay, what else?
2 nuclei.
Okay, there looks like there's 2 nuclei. What else? Compared to this one, okay, here your granules stained bluish, purplish, okay, what are these stained? Red, okay. Eosin is a red dye. So eosinophils love the red dye. The granules in an eosinophil pick up red dye. Okay, so you're going to see a ton of granules, and you're going to see a reddish coloration. Eosinophils are specialized for 2 things: taking out parasites and cleaning up after allergies. Taking out parasites and cleaning up after allergies. So sometimes you see increased eosinophils when somebody's having a flaming allergic reaction. Sometimes you will see increased eosinophils when somebody's having some kind of worm infection, for example. Okay. The next type of white blood cell is the basophil. What do you see there? What pops out at you? Okay, it's really dark. Okay, do you see granules? Yes, you do. Are they dense? Or yeah, they're really, really dense, and they're really, really dark. With eosinophils and with basophils both, but particularly with basophils, there can be so many granules that it almost looks like it obliterates the nucleus. Okay? But basophils are dark. Basophils secrete both heparin and histamine. And they are involved in inflammatory reactions. Now, we're going to see in micro, and also in a couple of weeks with the immune system, inflammatory reactions are designed to protect you from invaders. Okay? So you will see basophils elevated when you have an inflammatory reaction. Now sometimes allergic reactions are inflammatory as well, so you can see an elevation in basophils sometimes with allergies. Chanel?
You said heparin and what?
Histamine. Histamine.
So, if you were looking at like the breakdown and there was a rise in eosinophils but not in basophils, would that indicate possibly a parasitic?
Yeah, now you always look and see what else you have going on with your patient as well. Okay, there are leukemias, for example, that will increase eosinophils. Okay, but you know that's the direction it tips your thinking. Okay, you don't treat a blood test of any kind. Okay, you don't treat a medical test of any kind. You treat a person. Okay, which means that you need to see what else is going on with them. All right, monocytes. Monocytes and lymphocytes are agranulocytes. And now you can see why I get annoyed at that characterization. That monocyte looks like it has granules, doesn't it?
Yeah.
Okay. But what is the thing that pops out at you about that monocyte?
The
Large.
It's huge. If you look at the rest of these guys, the monocyte is much bigger, okay, and the nucleus is much bigger. Monocytes are called monocytes when you see them in the blood. Here you can see blood cells around, so you know that came from the blood. Monocytes don't necessarily stay in the blood. They crawl out of the blood, which is called diapedesis, and they crawl through the tissues. Okay? When they do that, they are called macrophages. Now, what is phagocytosis? Cell eating. Okay, so a macrophage would do what? Eat like crazy, eat tons. Okay, a macrophage is a walking garbage truck. Okay, it will clean up cellular debris. It will clean up tied up invaders. It will clean up antibodies. It will clean up damaged tissue. It will clean up bacteria. Okay, lots of things. Macrophages are important in the immune response. And then lastly here, you have the lymphocyte. Now what stands out at you about the lymphocyte? Number one, it's smaller, and number two, it looks like the nucleus takes up the whole cell. There's just a very thin rim of cytoplasm around that nucleus. Lymphocytes are specialized to take out abnormal cells. Lymphocytes are also specialized to produce antibodies. And we will get into the details of that down the road. So lymphocytes take out abnormal cells and they take out, or they produce antibodies. All right, that's where I'm going to stop for today. Next week we will continue on and we will talk about clotting, and we will talk about the blood typing and reactivity that blood cells exhibit. Okay. All right, you guys be sure and turn in your PhysioX